like to say thank you for inviting me to moderate this exciting session. So I'm at the National Human Genome Research Institute, and I think our keynote speaker and the previous speakers have emphasized that genomics influences everything. So while this particular area is not my field, I'm really happy to be able to introduce it. And so my division director asked me what session I'm moderating, and I said, I'm moderating the other session. And really, this is the other session, right? Because it's not about how sex influences us internally, but how sex allows us to interact differently and has us interact differently with external influences. And so our speakers are going to talk about actually a wide variety of these external influences. We'll hear about environment and lifestyle. We'll hear about treatments. We'll hear about vaccines. And at the end, how our sex influences our behavior, which in turn influences how we respond to our environment. And so without further ado, I am going to talk about our first speaker. And in doing so, I have to say that while one of the hats I wear is a program director at NHGRI, another hat I wear is one of the coordinators for one of the transformative Common Fund programs that Betsy mentioned earlier, and that's Human Heredity and Health in Africa, or H3 Africa. And we're very pleased to have one of our PIs from that initiative here. It's part of the Common Fund Global Health Initiative. So I think Michelle Ramsey gets the honor for having traveled the farthest to get here. She's here from the University of Witwatersrand in Johannesburg, South Africa, where she is director of the Sidney Brenner Institute for Molecular Biosciences and is in the Department of Human Genetics. So Michelle is going to talk to us about sex stratification for obesity and related traits in African populations as part of the H3 Africa A W Y study.
Thank you very much, Jennifer, and good morning to all of you. I must say that I'm really delighted to be part of this meeting and also to be one of the groups that is a beneficiary of this Common Fund initiative looking at sex differences. So it's a really great pleasure to be here. And I think what I find so interesting and exciting about this particular meeting is the cross-disciplinary interactions, and already I've learned so much this morning. I think that's what's part of the pleasure of coming to these meetings, so thank you so much for the opportunity of being here. So I'm going to talk a little bit about sex stratification and obesity. I must say this is one of the conversations we often have about whether sex should just be included as a confounder or whether we actually need to analyze our data differently, putting females in one group and males in another. So I've just put a few kind of headings where I just want to highlight some things about the work that we do. Our work is essentially about body composition and cardiometabolic disease risk. But what we do know is that there are huge differences in the distribution of obesity in many different ways, and these have challenges, I think, for sex as a biological variable. So there's sex differences in obesity relative to the country that you come from, relative to individual socioeconomic status or circumstance. There are differences depending on your ethnicity, and also we understand that there are differences according to sex. And I was quite interested to see one of the slides again showing that in many of the high-income countries, the difference between males and females in terms of obesity is very small. But when you look at low-income countries, middle-income countries, very often this difference between males and females in terms of obesity is quite large. So what is this? Is this about ethnicity, because there's also an ethnic split, or is it about culture, or is it about environment? So I put the slide up with trepidation, just having heard Margaret's talk. And I love the idea of a continuum, because I think there is a continuum in terms of differences. There are some very important differences, and I'm just having 2 very short slides about this. But I think men and women have different reproductive strategies. Of course they do. And that has to do with hormones a lot. But there's also a difference across the life course in terms of men and women and obesity. So when we think about obesity, we think about different body shapes. And part of this has to do with the fact that men, when they become obese, accumulate their fat in different places to women. So men are more likely to accumulate visceral fat, so fat around their organs, which has great danger in terms of cardiometabolic risk, whereas women tend to accumulate their fat more in subcutaneous areas. And this does not have the same deleterious effects during certain stages of their life. But post-menopause, things change again, and they then start accumulating more visceral fat, giving them greater predisposition to cardiometabolic disease. So as we increase our affluence, we kind of subjected to increased energy intake. And again, maybe we deal with this differently as men and women, but also differently during different stages of our development. And this can have the effect of either producing healthier fat or adipose tissue, or unhealthy, inflamed and fibrotic adipose tissue. And this has to do with many of these pathways that show differences again between men and women at different stages of their life. But I did want to pause here a little bit to look at the life course and look at, when we progress from in utero life all the way through to older adults, what are some of the sex differences that we see? So it was emphasized again this morning that already before birth there are important sex differences. But we know also that the environment is important, and this session is about the impact of the environment. And the mother's nutritional status, for example, can have very important influences on the outcome of adult obesity. In South Africa, when we look at stunting in the early infant, about 1 in 5 children in the ages of 1 to 4 are stunted in South Africa. But look at what happens as they progress through puberty, in that you then see a dramatic increase in obesity, particularly in females. Those are the grey bars as opposed to the males, who are the black bars. So with increasing age in our currently obesogenic environment in South Africa, teenage girls are really susceptible to becoming obese. When we then look in terms of reproductive years, there are very different strategies in terms of promoting obesity. And what you can see here is differences from, say, the teenage years right up to older age, in terms of differences in the percentage of obesity between men and women in South Africa. Now, South Africa, I have to tell you, is very far along the epidemiological transition of increasing urbanization, lifestyle changes, which is not the case for all African countries. And then we know as women progress past menopause, we get a convergence again in terms of how we react in terms of obesity and risk to cardiometabolic diseases. So now I want to just change a little bit and think about epidemiological perspectives in terms of obesity and cardiometabolic risk. So we know that there is a link between obesity that's been shown time and again in terms of increased risk for hypertension, diabetes, other cardiovascular diseases. But what we know too is that there are huge regional differences across the world in terms of how we look at obesity, and I'm going to show you some graphs just to show you what's happening in Africa. But we know that differences are because of some fixed factors like genetic sex, age, but also some factors that change enormously, like the environment. And I was when I was reading and thinking about this meeting, I was struck by the fact that we're not just looking at sex differences, we're looking at the influence of sex on some of the outcomes. So when we just compare here the US and South Africa in terms of obesity and what has been happening over a period of time, so we're going right back to the 60s, fast forwarding to 2012 in the US, and you can see this tremendous increase in obesity both in men and women. And when we look in South Africa, if we just look from 1998 to 2012, we can see that there has been an increase in obesity. But what is happening here is that women are really disproportionately affected by obesity in Africa than are men. And when we look at the correlation between the prevalence of obesity and the prevalence of diabetes, you can see that there is a positive correlation. And this is more so in both South Africa and in North Africa, like in Morocco and Egypt. But you can see that the majority of African countries have very low levels of obesity because of poverty and not the sort of exposure to high-calorie foods that are affordable. Then just another slide showing some data from South Africa in terms of individuals who are either, just want to get this, normal weight, overweight or obese, and then looking at some of these indices which increase their risk for comorbid diseases. And those are things like dysregulation of glucose metabolism, having high LDL cholesterol but lower HDL cholesterol, and increasing hypertension. So what is happening in Africa? So Africa is really experiencing this health or metabolic transition. And what is so important is that it's at different stages across different places in Africa. And one of the reasons we actually want to study this is because we have this opportunity at looking at the early, medium, and late stages of this epidemiological transition. And we really want to search for key drivers that inform interventions that will prove health going forward. So this is showing you a slide on the prevalence of overweight and showing you the difference between men and women over a period of almost 30 years. And again, what you can see here is that women are disproportionately affected by being overweight in terms of men in many different places. But I also want you to see this difference across the African continent, with South and North being more affected and further along this transition. So this brings me to our H3 Africa project called A W.Y, which stands for African WITS In-Depth Partnership. So WITS is our university. In-Depth is an international network of demographic health and surveillance centers, which is a fabulous platform to use to study anything, because those are communities that have been studied longitudinally, and also where it is possible to go back to participants if you want to find out what has happened to them. So, you know, if you want to look at cardiometabolic endpoints, you have this longitudinal view, which is very important. So A W.Y is about studying 12,000 older adults between the ages of 40 and 60, both males and females, and I'm going to tell you about some of our challenges there. We're looking at body composition, but we're also looking at cardiometabolic disease risk factors and outcomes. So we're really interested in understanding gene-environment interactions. We're very happy that we are now funded for a second phase, and this will allow us to do longitudinal studies, but also to look at the menopausal transition. And I think this is going to be important because many of the women that we've been studying will have been premenopausal at the beginning of the study and then will transition to postmenopausal state. So we'll have an opportunity to look at that. And again, this morning, I was interested in the whole story about the role of the microbiome. So this is one of the elements that we've built into the second part of our study. So this is a picture that I took on a visit to Burkina Faso, which is in West Africa. And this is one of our study sites. What you saw on the map previously were the 6 places that we are doing our study. And you can see here that there are 8 women and 1 man. And it seems in Africa, and I don't know if this is true for the rest of the world, but that women are more eager to participate in health-related research than are men. So our challenge is almost the other way around. And we've seen this particularly in one of our South African sites called Degale, which is a rural site in the north of South Africa. And there we were not able to achieve an equal male-female balance. And the reason there was that many of the men are migrant workers. And between the ages of 40 and 60, they would be leaving the rural areas to go and work in the cities. And if they did come home for holidays or for weekends, they didn't particularly want to participate in questionnaires and measurements and things. So our challenge has been to get enough men at some of our sites. So I just wanted to show you a little bit of our data in terms of cardiometabolic disease and then also in terms of obesity. So this is a slide looking at hypertension, and we have 6 sites. On the left are the 3 South African sites, then Nairobi in East Africa, and then 2 West African sites, Nanoro and Navrongo. And what I've shown there is the prevalence of hypertension. So you can see, again, this reflects this epidemiological transition in that South Africa has a much higher prevalence of hypertension than do the other parts of Africa. But what you can see here are also the male-female sex differences. So the women are shown in the orange, the men in the maroon. And you can see that in 3 of the sites, women are more likely to be hypertensive, but that in Nanoro, our Burkina Faso site, it's the other way around. So there's no consistent pattern, and we see this with quite a few different things. Then we decided to take everybody who was hypertensive and to put them into 3 groups and say we have hypertensives who are controlled, that's the dark maroon. Then we have hypertensives who know that they have high blood pressure, but they're not controlling it, that's the orange. And then there was the grey of people who were not aware that they had high blood pressure. So you can see across these different African regions, there are many, many people who are not aware of the fact that they have high blood pressure. But even of those who are aware, very few of them are controlling their blood pressure. And you can see again here that there are many differences with the men always shown on the left, the women on the right. But there are huge sex differences here as well. When we look at diabetes, there are also sex differences. And in our group of about 12,000 individuals, over 500 of them had diabetes because this was a population cross-sectional study. So this reflects what's happening in these communities. And in 3 of the sites, women were more likely to be diabetic than men, but in 2 of the sites, it was the other way around. So again, huge sex differences here. So let's look a little bit at obesity and at our 6 centers for different countries. Well, first of all, if we look at obesity reflected in this case as BMI, which we know is not always the best measure of obesity, we can see in men obesity generally is lower, but the variation is smaller, whereas in women, it's higher, but there's huge variability. So let's look at this in terms of women. So on the bottom here are the 3 South African sites. Then we've got East Africa here, and we have our 2 West African sites. So here we have not used BMI as a continuous variable, but have put it into 4 categories. So we have underweight, normal weight, overweight, and obese, obese being over a BMI of 30 and above. And what you can see here in terms of women is that in South Africa, in one of our sites, 67% of the women are obese. But when we look at West Africa, you can see how low the prevalence of obesity is. But there we have another problem, which is underweight. So this is what we see in terms of women. But what do we see in terms of men? So here the story looks quite different. So when you look at obesity, even in South Africa, there are not that many men who are obese, although there are more men in South Africa than in West and East Africa who are obese. And you can see one of the South African sites actually stands out here, and this is Tegale. And this is the site where we had a problem getting enough men. And I think that the men that we did get may have a different profile to what we might expect had we got a proper sampling there. So I think some really interesting things coming out here. So to summarize, we can say that BMI is higher in women than men, usually. Obesity is more common in Southern Africa than in East and West Africa, and this reflects the epidemiological transition. So what we wanted to do next is we wanted to look at obesity relative to a number of demographic, socioeconomic, and behavioral factors. And you can see here I've listed a whole bunch of them. And again, we decided to look at men and women differently. Now, it's such a rich data set that I can't even begin to unpack this, and I could talk for an hour just on what's happening here. But just for you to understand this very quickly is that when it's yellow, there's no data available. When it's green, there's no significant association between BMI and those factors. And when it's red, there is a significant association. So what you can see is that there's huge regional variability here in terms of the 6 sites across the top over here. But what you can see is that there are also differences between men and women. So we have a lot of work to do to try and unpack this, to understand the impact of the environment on obesity in these different regions in Africa. So we're busy now with applying hierarchical modeling to try and understand, first of all, the relationship between demographic data and socioeconomic status. Then we want to look at behavior, biology, and at this stage, the outcome is obesity. So we know through verbal autopsies that have been done in our centres that there is a high prevalence of mortality due to cardiometabolic disease, even in West Africa where obesity is not that common. We want to understand this better. There are huge regional differences in obesity prevalence. Women are more often obese than men, except for one of our centres. And then here I'm just going to give you a few vignettes from 3 of the centres in terms of some things that were really interesting to us. So Agincourt is one of our rural South African centres. And what we can see there is that, again, if we look at an increase in BMI, there are differences between men and women. So men who are married are more likely to be overweight, whereas women who are unmarried are more likely to be overweight. So there are differences there. And if we take into consideration all these different factors, they explain about 26% of the variance in men and only 14% of the variance in women. When we look at Nanoro, which is one of our West African sites, we can see very interesting differences there. But I think what has made me most excited about the work that we're doing there is that that population went through 2 periods of famine in 1974 and in 1985, and that would have been where some of our participants were either children or going through puberty, and I think that's going to be very interesting in terms of giving us a bit of a developmental perspective on what is happening there. And then Navrongo is one of our other centres in West Africa, and there what I'm most interested in is that they've seen very interesting differences in terms of ethnicity, because this particular rural site has 2 different ethnic groups, the Kasena and the Nankana, and they are showing significant differences, which to me is saying there's a genetic contribution here to obesity and how they behave. So this is my last slide, and this is just really talking about this epidemiological transition and the fact that we're going from more rural environment where people are getting lots of exercise to an urban environment where they're using taxis and they just have a high-calorie diet. So we know that there's an effect of urbanization on BMI, that there are rapid increases in non-communicable diseases. And as we hope we will see an increase in socio-demographic profile in Africa, this has to go together with a very important message, and that is educational programs on healthy eating and also on the dangers of obesity. So what we really want to understand now is also genetic risk for obesity, gene-environment interactions, and we probably need tailored health interventions. But do we also need sex-stratified interventions? So I think that's my message for today. And I did want to put this up. I know I'm going hugely over time, so just a few things to think about there, and to thank all the collaborators, because this is really a teamwork project. So thank you very much for your attention.
Very interesting talk, and I think we can take one question if somebody has one.
Nancy Lane, UC Davis. That was a great talk. I have a comment and a question, and I'll be brief so that we keep rolling along. One of the things I've noticed traveling through India last year is that, the different oils they use in different places and the contribution of that to diet and their increased incidence of heart disease. So my question to you is, have the fats changed that they're using in these different regions? Are they the same, number one, and also have they changed? Just curious.
Sorry, what is your question? Have the fats changed?
Diet.
The diet.
Okay, dietary
yes.
fat
Right.
intake, saturated fats that they cook with. Are they different between those regions?
So I don't know the answer to that. I know the diets are very different, so we need to investigate that much more thoroughly. And it's also changing.
Right.
Yeah.
Right.
So we need to look at that. So thank you.
So our next speaker, Dr. Anna Murphy, comes to us from the Neuroscience Institute at Georgia State University and is going to talk to us about sex differences in pain management.
Thank you very much for inviting me to be here. Which one is it? Which one? What do they call it? Laser. Oh, that one. Okay, perfect. Thanks. Perfect. Great. So thank you again for the invitation to give a talk here. I'm actually not funded through one of these supplements, so when Janine asked me, I wasn't really clear on what they were looking for. So I was like, sex is a biological variable. I'm not really sure this is appropriate, especially with this administration. And Janine's like, no. And I'm like, this? Pain and the influence of sex? And she's like, no. And so finally I was like, oh, okay, morphine, you want the influence of biological sex on how pain is modulated in preclinical and clinical studies. So I finally got it right. So thank you, Janine. So why study pain? Everybody's affected by pain. Over 20% of the population will experience chronic pain on a daily basis, and this is pain lasting approximately 3 weeks in duration. And remarkably, in 2017, opioids are still the #1 analgesic used for the alleviation of persistent pain. And surprisingly, very few studies have actually asked the question, does morphine produce a comparable level of pain relief in both males and females? Several studies have been done preclinically. These studies have traditionally used a rodent model, either my favorite, the rat, or the non-rat mouse. And these studies have primarily used a variety of different acute assays. The tail flick, which the animal sits here and a radiant beam of light hits its tail and it flicks it in about 7 seconds. The hot plate, or the formalin injection into the temporal mandibular joint. And for the most part, these studies have all produced mixed results. Sometimes morphine works better in a male, sometimes morphine works better in a female, but there's been a lot of variability in the results. Same thing with the experimental studies in humans. These are all like heat pain testing, cold pressor. This is why you shouldn't let your child post photos on the internet. And the isotonic saline injections. Again, sometimes morphine works better in a male, sometimes morphine works better in a female. But again, these are all very acute pain assays. And so then we've also looked at clinical studies, started looking at, well, what about analgesic consumption? How often do males and females push the patient-controlled analgesia, the morphine pump? And it turns out males actually produce the morphine, or will self-administer morphine more frequently than females do for post-surgical pain modulation. And this was interpreted for a long period of time that, well, morphine clearly works much better in females than males. Poor the males, they're still suffering, they're pushing the analgesic pump, and females are perfectly good to go. Well, it turns out it's very important to remember that consumption is not equivalent to analgesia, and it turns out that all the negative side effects associated with opiate consumption, including headache, dysphoria, vomiting, GI immotility, are exacerbated in females as opposed to males. So it's not that females are more analgesic, morphine's working that much better, it's that we're feeling sick and we would rather just bear the pain than continue to self-administer morphine. So we went back to our rodent model of pain and asked, what about chronic pain? If you inflict some sort of a injury on an animal, and we use complete Freud's adjuvant, which produces, it's an inflammatory response. It lasts the initial response is within 5 hours, and it lasts for about 3 weeks. If you use a chronic assay for pain, do you see differences in morphine's ability to modulate that pain? And this just shows you, yes, it hurts. It's about a 200% increase in paw diameter. It's an inflammatory pain. It's one of the most common clinically presented pain syndromes that there are. So this is showing you some of our brief data that if this is a latency to withdraw the paw in response to a noxious thermal stimulus following the CFA. And this is the dose of morphine. This was given systemically. Males typically have a 2-fold reduction in ED50 values, so females require twice the amount of morphine as a male to produce comparable levels of analgesia. These differences are not due to sex differences in baseline pain sensitivity. We typically, in all of our assays, do not see any differences in baseline pain sensitivity. They're not due to differences in the edema produced by the complete Freud's adjuvant into the hind paw. It's also not due to differences in the hyperalgesia produced by the CFA. and we did not see any effect of estrous cycle on any of the parameters examined. These differences are also not due to sex differences in the pharmacokinetics of morphine. Typically, you don't see any differences in the brain metabolism rates of morphine or serum levels of morphine. So then the question is, what is it about the central nervous system that is differentially organized in order for these differences in opiate analgesia in the chronic pain assay? There are several mechanisms that could contribute to some of these differences that have been observed. We've chosen to focus on the mu-opioid receptor. This is the preferred receptor for morphine. It's the obvious first place to look. Are there differences in how mu-opioid receptor transduces the effects of morphine? Are there differences in the receptor expression? Are there differences in the G-protein coupling, the intracellular signaling cascade, the GERCs, and so forth and so on? Straightforward question. You would think by 2017 somebody had asked this question, are there sex differences in mu-opiate receptor expression in the central nervous system? And remarkably, nobody's really looked at that. This is not an orphan receptor, I'd like to remind you. This is a receptor that has been implicated in pretty much every single physiological function you can think of: feeding, sex, drugs of reward, anything. The mu-opiate receptor has been implicated, but yet nobody's really looked to see if it's sexually dimorphic, or if there are sex differences, sorry, Peg, if there are sex differences in its expression throughout the central nervous system. The opiate receptor is highly promiscuous. It's brain, brainstem, spinal cord, periphery, GI system. This is how you see a number of the different side effects associated with opiate consumption. We've chosen to focus on what I'd like to consider the heart and soul of the central nervous system, the midbrain periaqueductal gray. This region of the brain located in the midbrain is critical for the analgesic effects of opiates, and pain relayed through the central nervous system through the primary afferents activates the PAG, which then releases endogenous opiates, which ultimately wind up blocking pain at the level of the spinal cord. In addition to your endogenous opiates, exogenous opiates produce a physiological action primarily through the PAG as well. So I can give you somebody morphine systemically, block the opiate receptors in the PAG, and morphine will be significantly attenuated. So it really is one of the meccas for opiate action in the central nervous system. So we asked the question, are there sex differences in mu-opiate receptor expression in the PAG? And sure enough, of course, there is. Males, this is using immunohistochemistry to look at the opiate receptor distribution in the PAG, and females are on the bottom. This is showing you the quantification, and males typically have significantly greater levels of mu-opiate receptor expression in the PAG, and that's showing you also, we've shown that with Western blots. So you have the sex difference in mu-opiate receptor expression in the PAG. We didn't see any sex differences elsewhere. We have, actually, there was one other region, the parabrachial nucleus, which is critical for visceral pain, and we think that's one of the reasons why females suffer from the preponderance of visceral pain disorders, or it contributes to it. So given these sex differences in mu-opiate receptor expression in the PAG, then it should follow that if you put morphine directly into the BAG, you're going to see a greater analgesic response in males than females. And sure enough, blue are boys, pink are females. It just works better that way. Everybody knows it. And these are for different doses of morphine. This is % maximum possible effect. And you can see morphine consistently produced a greater degree of analgesia in males. Females, nothing until you get to the much higher doses, and still it's not really all that effective. Just to hammer the message home, we said, well, is mu-opiate receptor expression in the PAG necessary and sufficient to drive this sex difference? So we used a compound called dermorphin saporin. Saporin's a ribosomal inactivating factor. It's conjugated to dermorphin, which is a preferred ligand for the mu-opiate receptor. So we can completely take out the opiate receptor in the PAG, and then ask the fact, what is the impact on opiate analgesia? And then, not surprising, males, you see a threefold increase in the ED50 value when you take out the opiate receptor in the PAG. And females, it absolutely had no effect. So it didn't matter if there were mu opiate receptors in the PAG or not. Morphine was still, it didn't really impact it. And by contrast, it had a huge impact on males. So again, suggesting that the opiate receptor in the PAG is a critical factor driving these sex differences in opiate analgesia. And this is just pointing it out one more time. So one of the other things we want to look at were, are there other mechanisms that are contributing to some of these sex differences besides just the opiate receptor? And about that time is when Mark Hutchinson and Linda Watkins published their seminal data showing that microglia, and in particular the microglia located toll-like receptor 4, has an amazing effect on opiate modulation of pain. Opiates like toll-like receptor 4, they bind to it and activates microglia, and resulting in potentially the attenuation of opiate analgesia. So we next asked the question, well, so here's the toll-like receptor 4. Again, it's found on the microglia, and it's a pattern-associated molecular pattern receptor, pathogen-associated molecular pattern receptor. Sorry, I have a cold. And so it has a number of preferred ligands, including lipopolysaccharide. Activation of the microglia in the toll-like receptor 4 initiates the production of a variety of pro-inflammatory cytokines, including interleukin-1 beta, IL-6, and TNF. The PAG, it turns out, has a ton of toll-like receptor 4 expression, and making it a perfect potential mechanism that would also help drive some of these observed sex differences in opiate analgesia. So we asked, what is the role of the microglia and toll-like receptor 4 and PAG, opiate-mediated analgesia? So morphine, again, just a schematic, binds through the toll-like receptor 4 as well as through the mu opiate receptor in the PAG to initiate pain modulation at the level of the RVM and then the spinal cord. And basically, morphine binding to the toll-like receptor 4 on microglia results in a release of TNF. TNF binds to the TNFR1 receptor. This results in a decrease in GABA receptor expression and an increase in AMPA receptor expression. It also results in a decrease in glutamate transporter expression. I'm not presenting that data, but it's all there in the neuropsychopharmacology paper. And ultimately, what it's doing is it's creating an increased neuroexcitability environment within the PAG. Now, morphine works by hyperpolarizing PAG neurons. Now you have increased neuroexcitability, so instead of getting that nice, strong hyperpolarization induced by morphine, which is critical for pain modulation, you're getting a much weaker hyperpolarization. And so then the question is, well, okay, that's all cool, and we've shown that this is one of the mechanisms that contributes to opiate tolerance and so forth, but now what role is all this playing in the PAG and sex differences? And specifically, are there sex differences in microglia expression and toll-like receptor 4 that could potentially contribute and drive some of these sex differences that we've observed in opiate analgesia? So, we used a variety of different techniques to look at this, and what we found was that if you look, like Peg had talked about, there's microglia come in several different morphological subtypes, from your quiescent to your highly activated. And so we looked at activated microglia in males and females. And I should preface this by saying we didn't see any differences in the colonization of microglia, so basic densitometry or poor undergrads doing the cell counts. There were no differences between microglia number between males and females. Rather, when we started looking at the activation profile, type 1 versus type 4, were there differences there? And sure enough, at baseline, it didn't matter what you did to the animals, handle them, saline, morphine, females typically had a higher degree or higher percentage of activated microglia than the males did. And then this is showing you the range of the values for the different microglia activation profile for the handled, saline, or morphine-treated animals. Well, we noticed that there was a a ton of variability in this data. It was amazing the number of how much variability that we noticed. And also, if you'll notice, there's a lot of variability in an animal's response to morphine. Anybody who's done behavioral research before, especially behavioral pharmacology, you know that you can't just give an animal an injection and it's going to behave perfectly. You're always going to see variability. So I was like, wow, this is pretty cool. My student was like, oh my God, they're so variable. And I'm like, no. Let's embrace this variability. Let's see what proportion of these differences here that we can account for by differences here. How cool would that be if we can predict an animal's response to morphine based on their microglia profile? And this is just showing you, again, the resting microglia, the type 4, the real pretty ones, and then the activated microglia. So what we found was that in females, you could, the R-square, 46% of the coefficient of determination. In males and females with the morphine ED50 versus the percent of activated microglia. Again, it's for being able to account for 46% of the variability was pretty huge, I thought. I was really excited about this. It was primarily limited to the PAG. We looked at a variety of other regions, and there were no other significant correlations between percent activated microglia and morphine ED50 values. So then the next question was, well, okay, so if this is what's driving or also driving the sex difference in opiate analgesia, is these activated microglia, then we should be able to block the microglia or make them less active, and that way you should actually potentiate morphine's ability to produce an analgesic response in females. And sure enough, that's exactly what we found. We used positive naloxone, which is a positive isomer that preferentially binds to toll-like receptor 4, and we were able to. This is a normal female. This is the normal male, and you can see the sex difference that we previously shown and others. And if you block toll-like receptor 4, you significantly push the ED50 significantly to the left, such that now females require half the amount of morphine as a male. Right now you should be saying, wow, holy shit, right? That's huge. Sorry about my language. But, right, so, and in males it didn't really have any impact at all. So a twofold reduction in the analgesic response. We also thought, well, this is pretty critical. You have a brain region, the PAG, heart and soul of the central nervous system. It's involved in a variety of different physiological functions. So here you have, in females, the more activated profile of microglia. What does that mean in terms of a variety of other physiological functions? So we gave these animals lipopolysaccharide, which is, again, the toll-like receptor 4 agonist and induced sickness behavior. We measured febrile response and febrile and sickness behavior using a scale, and also measured cytokine expression in the PAG. And what we found was that animals who received lipopolysaccharide, females, it was night and day. You didn't even need a microscope. You could see totally the difference between the microglia expression in the females versus the males following LPS activation. We didn't see any differences in febrile response, but what we did see was a huge difference in the percent or cytokine expression in the PAG for males and females, such as TNF, IL-1 beta, and were significantly increased, and IL-10, which is anti-inflammatory, was significantly decreased in females who received LPS. So what are the clinical, I'm wrapping up, I'm sorry. What are the clinical implications of some of this? And so in microglia, the more activated state, lower threshold to activate microglia, and the greater cytokine release, which has huge clinical implications, like Emmerin talked about. Is it any surprise now that you have these huge differences in cytokine levels following some sort of a pathogen that females suffer from a greater preponderance of autoimmune disorders and chronic pain syndromes? No surprise to me at all. And so we think that these sex differences are driving the sex differences in microglia as well as mu-opiate receptor are driving the sex differences in opiate analgesia. And so how can we use some of this information clinically? Well, positive naloxone is a very potent toll-like receptor 4 agonist or inhibitor. And so we'd like to see more money put into the use of positive naloxone as opposed to negative naloxone. as a potential adjuvant for opiates, where we can significantly reduce the analgesic dosage in females. Also, there's a variety of different biologics that are coming onto the market that selectively sequester soluble TNF. Some of your previous ones, like etanercept, those interfered with both the membrane-bound and the soluble TNF. You don't want to mess around with your membrane-bound. That's one of the things that's conferring immunity. So a lot of people who are taking these biologics developed demyelinating diseases, which is not going to work, obviously. So, but we have now several biologics that will selectively sequester the soluble TNF, and we think that these should, we'd like to see these further developed clinically. Oh, and there. Thank you.
Thank you so much. That was fascinating, and I'm sure people will chase you down at lunch to ask questions. All right, our next speaker comes to us from the University of Minnesota, where she'll be talking about sex differences in vaccine-induced immune response. So, Dr. Nicole Basta.
All right, well, good afternoon, everyone. Thank you so much to the organizers for the invitation to present here today. It's really an opportunity and an honor for me to be here. I've been really fortunate to have benefited from support of the NIH, both through an Early Independence Award, which I received as a grad student, and some of the research I'm talking about today was funded by that award, and through a supplement in the Sex and Gender Differences program. So in this presentation, I'm looking forward to sharing with you my perspective as an epidemiologist conducting population-based research to understand the impact of vaccines. And in particular, I'm going to focus on the role of sex differences in vaccine-induced immune responses. So to start, I'll summarize some of the key aims of the field of vaccine epidemiology for those who might not be familiar with it, and set the stage by highlighting the importance of considering sex as a biological variable in these types of studies. Then I'll share some specific examples from my research on the new meningococcal A vaccine known as wMenACWY, and some of the sex differences research that we're doing there. This vaccine has been recently introduced in the African meningitis belt, an area that has annual meningitis outbreaks every year, and now more than 300 million people have been vaccinated. And finally, I'll wrap up my talk by spending a few minutes examining some of the challenges and opportunities that we're experiencing in our research in the hopes that it will spark additional discussion. So my research in the field of vaccine epidemiology has three primary aims. The first is really to evaluate the effectiveness of vaccines and provide evidence about the breadth and duration of vaccine-induced immunity. To do this, I primarily design and implement longitudinal population-based studies that are intended to complement clinical trials, but also go beyond them by assessing vaccine impacts in real-world conditions. Second, I aim to investigate possible biological mechanisms to understand the drivers of heterogeneity and variation that we see in immune response and vaccine effectiveness. And finally, my research also focuses on assessing the impact of different vaccination programs themselves, including understanding patterns of vaccine uptake, vaccine hesitancy, and other social and behavioral factors that shape how vaccine acceptance is changing in populations over time. So the key question that was posed to us speakers is, why is it important to consider sex as a biological variable in our research? So first, I think it's important to note that in the type of clinical research that I do, NIH has mandated for many years that both men and women are included in those types of studies. And in my research and many other clinical studies, equal numbers of males and females are typically enrolled. But as we've already heard today, considering sex as a biological variable goes way beyond that. One of the ways to motivate considering sex in a broad range of research is to consider some basic central concepts from epidemiology. For instance, we might wish to quantify sex-specific differences themselves. We might be interested in comparing males and females for some difference in outcomes or exposures that they're experiencing. In addition, many studies are concerned with accounting for sex as a confounder. So as many of you probably know, a confounder is defined as a characteristic that is associated with both the exposure of interest and the outcome of interest. Failing to collect data about a confounder or failing to include it in the analysis can really lead to biased estimates of the relationship between a given exposure and an outcome, which can lead to incorrect conclusions and inference. In addition, it's really important to evaluate whether sex is also acting as an effect modifier, meaning that the direction and magnitude of the association under investigation differs within the strata defined by sex. Ignoring an effect modifier and only reporting aggregate results can really lead to incorrect assessments as well. And there are numerous examples where sex is known to be both an important confounder and an effect modifier. The approach that one takes might depend upon the aims of the study, the role of sex in the mechanism under investigation as well. Now, more specifically with regards to vaccination outcomes, a recent review summarized the literature to highlight the evidence about different dimensions along which sex and also gender have been considered in vaccines research. The authors noted several key studies on the role of sex hormones and genetics in particular in studies of vaccine efficacy. They also highlighted the importance of considering gender in addition to sex in studies with complex drivers, such as studies of the microbiome and vaccine response. And they've highlighted the role of gender in investigating vaccine-related behaviors, such as uptake of vaccines and reporting of adverse events. There's really a growing body of research in each of these areas, and there's many interesting areas that we could discuss today. But I want to focus on vaccine-induced immune responses. While there's been quite extensive preclinical research that has demonstrated sex-specific differences in immune system function and response to natural infection, fewer studies have assessed sex differences in vaccine-induced immune responses, and the evidence that we do have is quite mixed. There are unexplained differences seen by vaccine formulation, the number of doses given, the timing of doses, the age of vaccination, and other factors. There's a particular lack of evidence as to whether any of the differences that are observed are clinically meaningful as well. So a recent individual participant meta-analysis of several clinical trials, shown here in the forest plot, exhibits these mixed findings. The authors found no statistically significant sex differences among infants following vaccination with meningococcal C, Haemophilus influenza B, or tetanus toxoid vaccines. However, they found higher immune responses among female infants following vaccination with diphtheria vaccines, pneumococcal vaccines, and to the A W and Y component of meningococcal A, C, W, and Y vaccines. Now, these findings summarize some really important questions. First, whether these similar differences exist in other age groups. This was a study in infants only. Second, whether these differences are clinically meaningful in any way, for instance, suggesting the need for booster doses or to account for waning immunity. Third, what factors drive the differences observed. And finally, whether newly developed vaccines or newly introduced vaccines also exhibit the sex differences observed in the degree or duration of immunity they induce. This focus on new vaccines is particularly important because understanding variation in immune response for new vaccines can inform the design of optimal vaccination strategies and suggest the timing and number of doses that we should use. So several of these questions about variation in immune response have been central to my research on the impact of the new meningococcal A vaccines known as MenACWY. MenACWY is a meningococcal A polysaccharide tetanus toxoid protein conjugate vaccine that was developed by the Meningitis Vaccine Project. It was designed specifically to prevent the large-scale outbreaks of MENA that had been occurring annually in an area known as the African meningitis belt, highlighted here on the map. In 2010, Mali, Niger, and Burkina Faso were the first countries to introduce the vaccine, which was disseminated through a mass vaccination campaign that targeted everyone aged 1 to 29 years old. In 2012, with the support from the NIH Early Independence Award and through collaborations with the Center for Vaccine Development in Mali and Public Health England, I launched the MenACWY Antibody Persistence Study, the MAP study. This study aims to investigate the population-level changes in immunity following MenACWY mass vaccination campaigns. It's a prospective longitudinal study of a randomly selected sample of 800 residents in Bamako, Mali, who were aged 1 to 29 years at the time of vaccination and received the vaccine. The design of the study is that we conduct repeated serological surveys 2 years after vaccination and 3.5 years after vaccination. And our primary outcome has been MENA-specific serum bactericidal antibody titers, SBA titers. This is the accepted correlative protection that's used for licensing meningococcal vaccines. And our secondary outcome has been to look at tetanus toxoid IgG antibody levels, since the vaccine is also expected to boost tetanus immunity. So if we look at some of our key results, our primary aim was to assess the distribution of MEN-A immunity 2 years and 3.5 years after vaccination. And we found robust and persistent immune responses across all age and sex groups. We typically expect a putative protective threshold of 128, which is shown here by the dotted line, in settings where there's high background immunity. And regardless of age and sex, we see a high proportion of individuals maintained immunity in this range. However, we did note some variation by age and sex and the proportion of participants with titers at each level. So we then followed up by comparing sex-specific differences in our sample. And here we show the geometric mean titers for males and females, along with the 95% confidence intervals for each group by age. What we observed is evidence of higher wMenACWY titers among females less than 18 years across those age groups, and a suggestion of lower titers among older females, 18 and older. Similar trends persisted at 3.5 years after vaccination as well. This finding quantifies the sex-specific differences, but then the key question for us became, what might explain these trends observed? So in work currently being led by my PhD student, Maria Sundaram, she's investigating the role of potential drivers that might explain the variation in immune responses that we're observing. So based on a number of preclinical studies that suggested different mechanisms that could explain these differences, she's focusing her efforts on examining the role of micronutrient status in both chronic and acute inflammation in relation to immune response to MenACWY. So just to give you one example which highlights a point that I made earlier, I've included here one of Maria's analyses of the association between the micronutrient RBP, retinol binding protein, a marker of vitamin A status, and wMenACWY titers. The result of her initial linear regression model suggested no association between RBP and SBA titers after controlling for sex, age at vaccination, and markers of acute and chronic inflammation. But in a stratified analysis, she observed a trend towards a positive relationship between RBP and SBA among females, shown here in red, and a negative relationship among males, shown here in blue. This suggests that sex may be an important effect modifier in this context, and that by aggregating both sexes in this analysis, the differences were obscured. So in ongoing analyses, Maria is examining these associations and others in greater depth, primarily to generate hypotheses that could be investigated in future studies. So next, I wanted to share results from our secondary aim of the MAP study, and that was to investigate whether MenACWY could boost long-term tetanus immunity among vaccinees. So I noted that MenACWY is a meningococcal A vaccine that's conjugated to tetanus toxoid, and there was evidence of short-term boosting of tetanus immunity during clinical trials. What we found was that tetanus toxoid geometric mean concentrations remained significantly higher among all age groups 2 years after MenACWY had been introduced. And these tables show the increase from the pre-vaccination titers in the population to the 2-year post-vaccination titers for both males and females. This increase is still observed 3.5 years after vaccination, though the impact is lower due to some waning immunity. This is really an important finding in a region where tetanus and maternal and neonatal tetanus are still remaining quite a problem. So given this finding about the boosting of long-term tetanus immunity, we wanted to quantify the increase in the proportion of individuals with robust tetanus toxoid IgG, which would be indicative of long-term protection. Defined here as an antibody concentration greater than or equal to 1 international unit per milliliter. So what we see is that prior to MenACWY introduction, the dashed lines, both males and females had relatively low proportion of individuals with long-term protective titers, except for in the case of females aged 18 or older, where the proportion protected was quite high prior to MenACWY introduction. Following MenACWY introduction, the solid lines here, we see that the immunity to tetanus was boosted in all age groups, and that difference was significant. The higher baseline titers seen in females are really due to a targeted vaccination program, which encourages women of childbearing age to receive tetanus toxoid vaccine to prevent maternal and neonatal tetanus. And that reflects the impact of a vaccination program rather than any biological mechanism that is creating these differences observed. So to summarize, we observed trends towards higher MenA vaccine response among younger females, consistent with findings from a recent meta-analysis for other meningococcal vaccines. Our ongoing analyses are investigating possible mechanisms, but they suggest associations with micronutrients may differ within strata defined by sex, highlighting the importance of disaggregating and reporting sex-specific results. And finally, we've observed sex differences in trends in boosting of tetanus immunity, which really reflect country-specific vaccination program priorities targeting women of childbearing ages rather than any sort of biological mechanism. So to wrap up, I would like to just note a few challenges and opportunities that we've encountered in the hopes of sparking some discussion. So I think it's important to note that all of the results that I've shared today are the result of secondary analyses. They're all exploratory or hypothesis-generating analyses. Our study was powered to assess age group-specific differences only at the outset. So further advanced planning would be needed to include sex as a biological variable prior to data collection during the planning phase, because larger sample sizes are really going to be needed in order to verify some of the hypotheses that we're generating from these data. In addition, thoughtful and structured data analysis plans are really key to appropriately handling sex as a confounder or an effective modifier, based on the example that I showed you. And we also see a need for consistent reporting of sex-specific results and not just positive or significant differences in all clinical research. Because it's been very difficult to find other studies that have looked at sex-specific differences in clinical analyses of vaccines. Combined analyses generalize to a hypothetical individual of average sex, which doesn't really reflect any individual in the population, and that can obscure possibly meaningful sex-specific differences. And finally, we see a need as well for additional preclinical data on sex differences that could inform the design of clinical studies and motivate investigating novel mechanisms in population studies. We always look to preclinical studies to try to determine what mechanisms we'd like to investigate at the population level. So this has been really a rich source of ideas. So with that, I'll wrap up this talk and just thank my collaborators and the students in my group and the NIH for the funding that has made this research possible. Thank you.
So Nicole left us some time for questions, if anybody has any.
Morning, thank you. With regard to the differential titers against the meningococci that you identify in the females and males, you've got pretty high titers. They're like sort of twofold differences. Is there a way to model the efficacy of those titers or those titer differences in neutralizing the organism?
That's a really good question. It really highlights the point that I made about we're not sure if any of the differences that we see in vaccine-induced immunity are clinically relevant, especially in the context of the African meningitis bout, where you have pretty high pre-vaccination titers due to the circulation of carriage strains. It's not clear that having a higher titer response for females will make any difference in terms of their protection going on. What we think it might indicate is slower waning of immunity over time and perhaps a longer duration of immunity.
Hi, very interesting information. David Hunstead, Pediatric Infectious Diseases at WashU. My question is about, there's a lot of data in the literature about females having augmented responses to various vaccinations, and we always think about the main antigens, right, MenA or tetanus toxoid, but I'm interested in whether there are any data or whether you have comments about how we could look at sex-specific development of vaccines as regard the adjuvants that go in there. We've heard about TOL4, which is a target of some adjuvants that are coming out now. And so are there data out there about sex-specific differences according to the adjuvant rather than the primary antigen?
That's a great question. I'm not familiar with any studies that have reported sex-specific differences with respect to the adjuvant that's used in the vaccine. There's certainly a number of studies that have looked at reported side effects in terms of their sex-specific differences and have found that females experience and report side effects much more commonly than males do across all age groups.
Thank you. So our next speaker is one of the talks chosen to be elevated from a poster to a talk. And Dr. Catherine A.W.Y is going to, I think, continue some of what Dr. McCarthy had tantalized us with earlier. And while I'm sure the talk is excellent, I feel like this was chosen to challenge my ability to say a title. I'm going to do my best. Oh, you have a shortened version. Okay. And endocannabinoidals drive development of sex differences in social play.
All right, thank you.
So I'd like to thank the organizers for the opportunity to speak. As was mentioned, I will also have a poster, so if anyone is looking for more detail, please stop by. The specific type of social behavior that I will be talking about is this rough and tumble play behavior, which is also commonly referred to as play fighting. It's seen across almost all mammalian species and is absolutely critical for normal brain maturation during the juvenile period. And also across almost all mammalian species, with a few very rare exceptions, it is more frequent and intense in juvenile males relative to females. So we study juvenile rats, and this is a figure from rats showing in the blue a male-male pair, and you can see very nicely compared to the females in pink, higher levels of play events relative to the females. I also want to point out one of the things that I really love about this behavior here, and that is that if you pair a male and female with one another, they go through a bit of adjustment so that the male will bring his behavior down a little bit to match that of the female, while the female raises up hers just a little bit. So it requires a lot of cooperation and communication between the animals to reach appropriate play behavior. Michael Meany and Bruce McEwen demonstrated that the sex difference in this behavior in rats is mediated by the medial amygdala, such that if you implant a testosterone capsule into a newborn female's medial amygdala, you see a dose-dependent increase in her play behavior, such that it will begin to reach the levels seen in males. I'm going to be talking about how the endocannabinoid system modulates this behavior, and so I just want to point out a few of the key components that I will be mentioning. And the first is 2-AG, or 2-arachidonyl glycerol, which is the most abundant ligand for the endocannabinoid system in the brain. And it is going to exert its actions primarily through acting on the CB1 and the CB2 type receptors. I want to point out that this system is one of the earliest turned on during development. The components can be seen as early as embryonic day 8, and suffice it to say that just about every component of brain development that you can think of, the endocannabinoid system has been implicated. So this is proliferation, differentiation, migration, and communication between almost all of the cell types within the brain. We have seen in the rat, if we dissect out the amygdala on postnatal day 4, males have a higher level of 2-arachidonyl glycerol relative to females. And if you inject females on the day of birth and the day after with testosterone, you get a very nice masculinization of her 2-AG levels, and this can be blocked if you co-administer an androgen receptor antagonist along with the testosterone. Knowing that there is this sex difference in the levels of 2-AG, we wanted to ask what happens if we exogenously activate a female's endocannabinoid system during this early period. So right after birth, females are given agonists for the CB1 and CB2 receptor, and I'm just showing here WHIN acts on both the CB1 and CB2 receptor, and this is a specific ACEA CB1 agonist given in a cocktail with a CB2 specific agonist, and you get a very nice increase in her play behavior. If you give these same treatments to the males, there is absolutely no change in his play behavior. However, the opposite is true, where if you give antagonists for the CB1 and the CB2 receptors to males, you get a decrease or feminization of his play behavior, whereas in females, this treatment will have absolutely no effect. So we know that higher testosterone in males increases endocannabinoids, such as 2-AG in the amygdala, which results in increased play. So you would predict following this that inhibition of the endocannabinoid receptors should block the testosterone-induced elevation in play. And very nicely, it does. We see our sex difference. If you compare the females and the males on the outermost 2 bars, you get a very nice increase in female play behavior when she is given testosterone, and you can completely block this increase if you administer endocannabinoid receptor antagonist. Again, this was the same antagonist that were used to decrease male play behavior on the previous slide. We know males have higher testosterone levels at birth, resulting in these higher 2-AG levels, and that females have almost no circulating testosterone, and thus lower 2AG levels. And this somehow mysteriously leads to these differences in play behavior once they're juveniles. Knowing that the endocannabinoid system acts on several different components of brain development, we wanted to see what was occurring at a more anatomical level in response to this difference. And this takes me to, I apologize for a sort of busy slide, but I will hopefully explain it. We have a sex difference in the number of newly proliferated cells in the neonatal amygdala. So this tissue was collected at postnatal day 4. That was the same age at which we saw the sex difference in levels of 2AG. And you can see male proliferation is lower than that of female proliferation. And I'm showing this by 2 different methods. We've seen it across several different experiments. In this top graph, we're looking at Ki67, a marker of pretty much everything except for a resting state cell, so not in G0. And it was quantified by flow cytometry, and we see a nice sex difference here. And then also, if we do immunohistochemical labeling for BRDU, a marker of newly synthesized DNA, we also see the sex difference. In the top panel, you can see that you can masculinize the numbers of newly proliferated cells with estradiol, dihydrotestosterone, which is a non-aromatizable version of testosterone, or testosterone itself. And down here you see that you can also masculinize numbers of newly proliferating cells with either a CB1 or a CB2 specific agonist. A sex difference in numbers of newly born cells can be due to differential proliferation or survival. So this takes us to the beautiful microglial cells, which Peg gave a very nice introduction to. These are the immune cells of the brain. I do want to point out that I am not the postdoc who ignored counting these cups here. The microglia is shown in this cyan color, and you can see it is wrapping this phagocytic cup around a PCNA positive cell, which is another one of our markers for newly proliferating cells. In the medial amygdala, the difference in numbers of phagocytic microglia correlates very nicely with the number of newly proliferated cells measured by BRDU. The cell slide is a little bit complicated, but what we did is blocked a receptor that is important for the ability of microglia to phagocytose any sort of a cell, not just limited to newborn cells. And this is within animal design. So one hemisphere, they received an injection of vehicle, and in the opposite hemisphere, they received an injection of this anti-CD11B, and it was directly into the amygdala. And you can see not much of a difference in females, but in males, you get a whopping increase in the number of newly proliferated cells when you block the ability of the microglia to phagocytose them. So this really tells us that without the action of the microglia, these cells would go on to survive just fine. So it is, in fact, cell murder by the microglia that is sexually differentiated in the amygdala. And one last thing, the sex difference in the numbers of phagocytic microglia is also regulated by steroid hormones. So up in this top panel, we again see a sex difference in the number of phagocytic microglia. This is exact opposite of what we see the proliferating cells. So it is lower in females compared to males, whereas proliferating cells are higher in females compared to males. If you give females testosterone, you increase their numbers of phagocytic microglia. And down here, we see that we also see this increase in the numbers of female phagocytic microglia in response to a CB1 or a CB2 specific agonist, along with the cocktail administration that we use to masculinize female juvenile social play behavior. So this is where we are at this point. We know that at birth, males have higher levels of testosterone, higher levels of 2-arachidonylglycerol. Those are correspondingly lower in females. And it goes along with males having higher numbers of phagocytic microglia and hence phagoapoptosis of newborn cells, resulting in lower numbers of newborn cells. But what we don't know is what leads to this difference in play behavior from here to here. So what we're currently working on is trying to actively inhibit phagocytosis in the males and seeing if we can affect their social play behavior in that manner. So with that, I'm going to thank the McCarthy Lab and members past and present. I'd like to thank Matt Hill at the University of Calgary, who did the mass spec work for us to measure 2AG, and of course our wonderful funding sources here at NIH. And hopefully there's a few moments for questions, but if not, please come to my poster. Okay. Oh, good. I was on time.
That was very interesting, and I think I followed most of it, but there was one slide that caught my attention. When you were trying to give the different hormones to match the male response, I noticed that you saw the same response with estrogen, DHEA, and testosterone. What's that about?
We're a little stymied by that as well, and it gets a little bit more complicated, which I will get into since you asked. The increase in 2AG. Oh, sorry.
Is that working?
Or I can just stand closer to
Stand
here.
over
I
here.
stepped out. The increase in 2AG that we saw, it seems to require an action of both testosterone and estradiol, such that if we give dihydrotestosterone, which cannot be aromatized to estradiol, we do not see that increase. So there seems to be this combined action in the amygdala where you need both testosterone and estradiol for full masculinization. As to why you would see the same effect on numbers of newly proliferating cells, I'm not sure. They could be acting on different aspects of regulating cell proliferation. Steroid hormones are known to provide a lot of trophic support for newly proliferating cells, so it's possible it's not 100% associated with the phagocytic microglia that I mentioned, but could be another mechanism.
All right, I'd like to thank all of our speakers. That was an excellent session. And do you have any announcements as we go to lunch?
Thank you so much. So we'll come back at 1:45 for the next session. So right now it's a lunch break. So if you need any directions to go anywhere, please let us know. Otherwise, we'll reconvene at 1:45. Thank you so much.